Today I am going to talk about the different structures of protein from primary structure to quaternary one. So here suppose we have a few of amino acids with different R group that are going to get involved into a condensation reaction that is done with the help of the removal of water molecules or say nucleophilic acyl substitution reaction to form peptide linkage or amide linkage. As here we can see, after the amino acid residues combine, it gives a amide group. It leads to the formation of a amide group, as we can see, that is involved in the peptide bond CO and H. This peptide bond also possesses a partial double bond character due to the lone pair of electron present on nitrogen, and this lone pair of electrons of nitrogen creates a resonance effect in which the electrons shared between carbonyl oxygen and amide nitrogen, thus it creating a partial negative charge on oxygen and partial positive charge on nitrogen. This ultimately leads to the formation of the peptide bond, and the backbone of polypeptide chain can be pictured in as a series of rigid plane, and this rigid peptide bond limit the range of conformation possible for a polypeptide chain because it is rigid. So here suppose we have the same structure with different amino acids bonded by a peptide link presented in pink color. So rotation around these peptide bond is not that much feasible because it have a partial double bond character. But there are some single bonds are present in this structure around which the rotation can be possible, and these angles are called as torsion angle or dihedral angle and are presented as phi and psi. Phi is the angle between the nitrogen and the alpha carbon, while psi is the angle between alpha carbon and the carbonyl carbon. So rotation around these angles is can be possible within the range of minus 180 degree to plus 180 degree, and this rotation leads to the conformational changes in the primary structure of protein. A linear polypeptide chain uh, may get alter on the basis of the rotation takes place around phi and psi. This leads to the formation of different a curvy structure, different kind of conformation, or the disturbance of atoms between the disturbance of the atom that is present around the peptide bond leads to different conformation of a linear polypeptide chain. Suppose here we have the same structure of the previous one, and here are the amino acids present within the structure. There are 4 amino acids are presented in this structure. So once the primary structure get altered on the basis of the dihedral angle changes, the atoms comes closer and it forms a hydrogen bond between hydrogen of nitrogen and oxygen of carbonyl group of first and fourth amino acid. As we can see the series first and the fourth one. Usually this kind of a structure also possesses a series of hydrogen bond in adjacent turn, and such a structure is called as alpha helix. One of the secondary structure in which each successive turn of the alpha helix is held to the adjacent turn via 3 to 4 hydrogen bonds. Thus, for the secondary structure to be formed, the NH group, this amino group, is the most important one, which is found in almost each and every amino acid, but in case of proline, it is present as a part of rigid ring. Thus, in such kind of conformation, proline cannot be found, as its presence may cause discontinuity in the structure because it is unable to form these hydrogen bonds. Another one structure from secondary structure of protein is Vita Conformation, in which the backbone of polypeptide chain is extended and is present in a zigzag manner rather than a helical one. So, hydrogen bond forms between the adjacent adjacent strands. Also, one more important thing, that is the R group. As we can see from this figure, R group in alpha helix, also in beta conformation, are protruded out from the structure. So, the beta structure or beta conformation can be found in two different ways. One is parallel, another one is anti-parallel, that I have explained in my previous video. Here I am presenting only the anti-parallel one because this one is the most stabilized beta conformation because of the collinear hydrogen bonding. Another one structure from the secondary structure of protein is beta turn. These beta turn are mainly found in globular protein and are present as a connecting element that links successive runs of alpha helix and beta sheets. These beta turn are rich in proline and glycine residue, as small R group of glycine is highly flexible and amino N of proline can easily readily assume the cis configuration. So this is all about the secondary structure that mainly form due to hydrogen bonding, or it can say the bond that is formed via between the peptide group. Now we'll move toward the higher category of the structure of protein, in which first one is the tertiary structure. Tertiary structure is mainly formed via non-covalent bond between the amino acid of the same protein. Within these bonds are intra molecular, which include the different category like yeah, first one is like ionic bond, dipole dipole interaction, hydrophobic interaction, and disulfide bond. Disulfide bond belong to the covalent one. So all non-covalent and the covalent bond are involved in the tertiary structure formation. Next is the quaternary structure. Quaternary structure is a complex. It's not a single protein or a single polypeptide chain. It's a complex of different polypeptide chain. As we can see from this diagram, 4 different chains are involved in the formation of a single biologically functional structure, which is a hemoglobin protein. These red color circles are presenting the heme group, that is a non-protein group, or can say prosthetic group. So quaternary structure is also possesses different bonds of both category covalent and non-covalent one. But the major feature of this quaternary structure that it possesses different polypeptide chain that are combined together to form a biologically functional unit. So this is all about the different kind of structure of protein. If you like this video, please hit the like button, share it, and also mention some comments whether it is negative or positive, so that it motivates me and make me to work accordingly. Thank you.